Hello, everyone. Welcome to today's live broadcast, Improved Understanding of Rainbow Trout Growth Physiology Through Functional Genomics. I am Christina Mahalik of Labroots.com, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots and sponsored by SciEx. SciEx's global leadership and world-class service and support in the capillary electrophoresis and liquid chromatography mass spectrometry industry have made it a trusted partner to thousands of scientists and lab analysts worldwide who are focused on basic research, drug discovery and development, food and environmental testing, and forensics and clinical research. For more information, visit SciEx.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button located in the lower left corner of the window. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page, and we'll follow the process for obtaining your credits. I would now like to introduce today's speaker, Beth Cleveland, PhD, growth physiologist, USDA, National Center for Cool and Cold Water Aquaculture. Dr. Beth Cleveland is a growth physiologist with the Agriculture Research Service at the USDA National Center for Cool and Cold Water Aquaculture in Kearneysville, West Virginia. Her research defines mechanisms that regulate growth and nutrient partitioning in rainbow trout with a particular emphasis on identifying endocrine factors that affect protein accretion in skeletal muscle. Recent work has utilized functional genomics and genetic modification to define the roles of specific proteins as regulators of nutrient deposition. Dr. Cleveland received her PhD in animal and food science from West Virginia University, where her research focused on amino acid biochemistry. I will now turn it over to Dr. Cleveland for her presentation. Dr. Cleveland, the floor is yours.
Thanks, Christina. The title of my talk is Improved Understanding of Rainbow Trout Growth Physiology Through Functional Genomics. First, I'd like to just give you an outline of the presentation. First, I will give you a brief introduction of myself and the research facility, and also give you a little bit of background on some of the research that we do here. But more time will be spent discussing some of the gene expression profiling that we've completed using the GeXP platform, specifically focusing on assay development as well as how we've applied these assays in different studies utilizing rainbow trout. And then finally, I'll finish up with some recent work on CRISPR-induced gene mutagenesis, particularly how we've analyzed genotypes using the GeXP platform. So I had forgotten that I had supplied a biography, so this will be a very quick review. I got my graduate degrees at West Virginia University in Animal and Food Science, and I started at the National Center for Cool and Cold Water Aquaculture in 2007 as a postdoc. And I was in that position for about a year and a half before transitioning over to the growth physiologist position, where I currently am today. This is a picture, an aerial photograph of our facility. It was built in about 2001, so it's still considered fairly new. The labs are quite large, quite spacious, so these are some images of our analytical labs, but we also have some considerable wet lab capacity as well. We can raise fish from egg stage through spawning because we have tanks of very different sizes. And our mission is to support and enhance the nation's cool and cold water aquaculture production through research and technology transfer. We have two major research areas. The first is genetics, genomics, and physiology, and the second is fish health and disease. The purpose of our facility is to perform research that has positive impacts on stakeholders of the United States and global rainbow trout industry. So this includes research that's pertinent to producers, feed manufacturers, processors, or, for example, vaccine developers, just to name a few. My research fits within the Genomics, Genetics, and Physiology Research Unit. The title of our 5-year research project is Integrating Research Approaches for Improving Production Efficiency in Salmonids. Central to this effort is our selective breeding program, in which we select for growth and fillet yield or disease resistance. The physiology-related research is important to this effort as we define phenotypes and additional mechanisms that are controlling these these economically important traits. The genomics research is both improving the current rainbow trout genome assembly, but more importantly, identifying DNA markers that will help us work towards genome selection programs. Central to physiology is understanding regulations of multiple systems, because this is really how the fish maintains normal function. Part of this is understanding what happens both within and between different systems or different tissue types to allow maintenance of homeostasis. This physiology-related research enables us to characterize things like how the fish responds to environmental stressors and how these stressors affect growth or, for example, the susceptibility to disease or reproductive success. This requires coordination across multiple systems, and so analyzing the proteins that are involved in the endocrine and autocrine responses are important for providing a comprehensive picture of the physiological response itself. Central to this is measuring changes in gene expression, because this information tells us that not only is the mechanism under regulation, but it is suggestive of increases or decreases in functional capacity. So collectively, gene expression profiling of multiple tissues is indicative of the coordinated physiological response. At this point, there are numerous methods available to measure gene expression. Some of the more common methods and the methods that we have available to us at Cool and Cold Water include real-time RT-PCR, microarray analysis, GeXP, and RNA-seq. Most applications have advantages and disadvantages, and these are certainly fit within that class. Real-time PCR, for example, while it is highly sensitive, cheap, and really at this point is routine in most labs, can be considered cumbersome because it's really only one gene per reaction. Sometimes there's multiplexing, but it usually only goes to a maximum of 3 genes per reaction. So real-time PCR can be considered low throughput and get quite expensive if numerous genes are required for many samples. Microarray analysis, while it can analyze tens of thousands of transcripts in a single sample, is also considered low throughput because it is costly. One is also limited only to analyzing genes that are on the array, and the sensitivity is a bit lower than what we can achieve with real-time PCR. With the introduction of RNA-seq, the value of transcriptomic analysis is realized, but it is quite expensive, and it requires a certain or a special expertise and computing capacity to analyze the large amount of data that's produced. This kind of approach can also be difficult in minor species like rainbow trout that does not have a fully characterized genome yet, and it's challenging to assemble the transcripts into meaningful data without losing a large percentage of the sequencing data. So a fourth approach that we have heavily utilized recently is the multiplex RT-PCR using the GeXP platform. The per reaction cost is similar to what we can achieve with real-time PCR. And the assays are developed to analyze usually between 25 and 40 genes per reaction. The experimental procedure is simple and enables high throughput, and it has levels of sensitivity that are between real-time PCR and microarray analysis. Some of the disadvantages of this technique is that the range of detection is slightly more limited than what is possible with real-time. There is some considerable optimization of the multiplex assay itself, And like the microarray, you're really limited to the genes that are within the assay. So today my talk will highlight how we've used the GeXP technology to further our knowledge of rainbow trout physiology through gene expression profiling. The multiplex PCR uses routine protocols and thermocyclers, along with SciEx reagents and primers designed and purchased by the researcher. The first step is a standard reverse transcriptase using a gene-specific reverse primer, followed by a PCR containing gene-specific forward primers. Each primer contains a universal tag sequence that enables amplification by universal primers in the PCR reaction. The PCR itself is a standard endpoint PCR, and the amplicons, or each gene product, is separated by capillary electrophoresis. One of the universal primers has a fluorescent tag, and so this enables amplicon detection. The data is extrapolated from a chromatogram, much like you see on the right of the screen, with each peak representing a single amplicon, or essentially a single gene product. The height of the peaks, or area under the curve, is directly proportional to transcript abundance. The overall premise behind the assay is that the primers are designed so that the PCR products differ by a minimum of 5 base pairs. This allows for adequate product separation and quantification using the capillary electrophoresis. As I mentioned before, an assay typically contains between 25 and 40 amplicons per assay, although if you're really careful, you could probably hit 50 genes per assay. This chart describes our general workflow from gene selection to assay design to using these assays in physiology studies. So at the Cool and Cold Water, we have designed and published data collected from a total of 4 multiplex, although today I'm really only going to be presenting data from 3 of the multiplexes. The 4th multiplex, which is not shown here, was really a collaborative effort and focuses on immune-related genes. But these 3 multiplexes are related to growth physiology, and they were developed to complement each other, so collectively we can generate a comprehensive picture of how endocrine and autocrine mechanisms are regulated to affect growth and nutrient metabolism. And this information can be coordinated with physiological responses like fillet yield or growth rates to characterize mechanisms associated with nutrient partitioning. So we have the 3 multiplexes shown here. One is related to growth mechanisms. The second is related to protein turnover, mostly protein degradation. And the nutrient or lipid metabolism multiplex looks at various genes associated with signaling pathways as well as lipid metabolism. Each multiplex contains the same 4 reference genes. So collectively, we have about 85 genes that are associated with growth and nutrient partitioning. There are likely 3 main tissues that are most significant for nutrient partitioning. The blue arrows in this chart represent the flow of nutrients from each compartment. The muscle represents 50% of the weight of the fish and is the final meat product. Increasing fillet yield is important to producers, and this is one of the focuses of our breeding program, so understanding mechanisms of muscle growth is critical. The liver is a central organ that produces hormones with systemic effects, such as insulin-like growth factor. It's also central for nutrient metabolism, and it largely represents regulates what nutrients are sent into peripheral circulation. The adipose tissue is also significant, and especially in salmonids like the rainbow trout. Adipose tissue accumulates in the abdominal cavity and can represent 75% of total body lipids and can reach about 20% of total body weight. While some of the lipids can be reclaimed by post-harvest processing, it's generally considered a waste tissue, or at least is much less valuable than the fillet. So increasing lipid deposition in the fillet rather than the visceral adipose tissue is certainly desirable. The growth multiplex is important in muscle and liver. One of the more valuable features of this multiplex is the inclusion of critical components of the growth hormone IGF axis. Plasma IGF is largely of hepatic origin, so measuring expression of this gene gives us insight into the regulation of one of the most central regulators of growth. However, about 97% of IGF in plasma is bound to IGF binding proteins, and it's thought that these binding proteins help direct IGF to specific tissues through their affinities to extracellular proteins specific for different tissue types. We know that there are 6 families of binding proteins, and each family has multiple pair logs. Some pair logs have autocrine functions, while others have endocrine functions, but we designed the multiplex to be fairly comprehensive for most of these binding protein family members. The growth assay also includes growth hormone receptors, which are critical for regulating IGF expression in liver, and are probably important for certain responses in muscle as well. The growth multiplex also covers myogenic regulatory proteins in muscle, and these are the proteins and transcription factors critical for regulating the proliferation and differentiation of myogenic precursor cells, which largely dictate muscle growth in indeterminate growing species like rainbow trout. Now, muscle growth is a function of both the rate of protein synthesis and the rate of protein degradation. The growth multiplex that I just described is largely descriptive for changes in protein synthesis. The protein degradation assay provides the other half of the equation, providing information on regulation of proteolytic pathways responsible for muscle protein breakdown. There are 3 main proteolytic pathways, all of which are included in the assay, with the addition of several caspases to provide information on apoptosis. The third multiplex covers mechanisms associated with lipid metabolism and lipid binding. Rainbow trout have a fairly low tolerance to dietary carbohydrates, and so dietary lipid content often exceeds 20% to provide this rich source of energy. Rainbow trout also deposit excess energy as visceral fat, so understanding lipid metabolism is key for determining whether the fish is in a state of nutrient deposition or nutrient mobilization. Changes in lipid metabolism are therefore suggestive of the metabolic state of the fish because they are largely based on the cellular ATP to AMP ratio. Many of those previously mentioned pathways are regulated by intracellular signaling, so we have developed the multiplex to contain several of the proteins that are important for these signaling mechanisms. An example are the TGF-beta superfamily members, which are important for growth factor signaling, including some components like myostatin and follistatin, which directly affect myogenic mechanisms. Signaling largely occurs through SMAD proteins, and the SMAD proteins themselves are predominantly post-translationally regulated by phosphorylation state, and this is what changes the activity level of these proteins. So while changes in gene expression are not indicative of changes in activity, they do suggest an increase or decrease in signaling capacity. So these genes are also included in the multiplex, such as the SMADs, the TGF-beta receptors, BAMB, follistatin, and myostatin. One more class of genes within the lipid multiplex are transcription factors important for regulation of metabolic genes. Examples are the PPAR transcription factors that are known to have gene targets involved in nutrient metabolism, such as fatty acid metabolism or glucose homeostasis. So measuring changes in expression of the PPARs will provide information on how the genes downstream of those transcription factors are regulated. So this approach enables us to hypothesize on many additional genes beyond just those that are included in the multiplex. Once one or more suites of genes are identified, then optimizing these genes or these assays is the next step. Much of the effort during optimization is spent quenching signals from high expressors, which in turn boosts signals from low expressors. This can be challenging, and there are some genes that are very low expressors that sometimes cannot be detected using these multiplex assays. This can be challenging for those genes, for example, some related to immune function, where responses are often an all or none response, where expression increases several hundredfold during a disease challenge. Baseline levels may not be detected, which can introduce challenges when analyzing and reporting data. Luckily, physiology-related genes don't have this problem, and so because they're not faced with this level of regulation, so generally all of the genes that we chose were easily detectable using this approach. So one of the first steps, as with nearly all PCR-based assays, is confirmation of amplicon identity. It's important to determine that the size of the product is the expected size. It's also important to determine the fold range of detection for each gene within the assay. Now, for my genes of interest, we stayed within the linear range of detection so that we could easily detect both increases and decreases in expression by interpolating data from a standard curve. This approach ensures the greatest accuracy and sensitivity for detecting changes in expression, since in physiology we rarely see increases that exceed tenfold. However, there are some situations where increases in expression can reach 100 to 500-fold, for example, in immune-related genes. And an extended standard curve is required to accurately quantify this response. In this situation, standard curves follow a third-order polynomial. And so knowledge of the expected expression response for a particular suite of genes is certainly helpful to determine which of these standard curve approaches is most appropriate. At the National Center for Cool and Cold Water, we have used the multiplex assay to characterize gene expression profiles for both in vitro studies, where we're isolating specific effects of treatments in one tissue type, to in vivo studies where we are interested in a systemic response coordinated across multiple tissues. So today my plan is to briefly highlight some of our findings within 3 studies that determine physiological mechanisms that are associated with 3 of these studies, including different growth trajectories, the significance of sex steroids for nutrient repartitioning during sexual maturation, and whether dietary phytoestrogens that can result from soy intake mimic estrogenic effects. We've also used these assays to look at gene dosage versus gene compensation in diploids versus triploids, although that information I won't be covering today. The first study I want to highlight is the compensatory growth phenomenon that is often observed in fish. The compensatory growth trajectory follows a period of feed deprivation or feed restriction that is often characterized by weight loss. And the red bar here indicates the period of feed deprivation and the weight loss that occurs. Once we start refeeding and going into the green growth stanza, there's a period of overconsumption and growth rates that exceed that of continually fed fish in terms of both growth rate and feed efficiency. Sometimes this growth response will shoot body weights above the continually fed group. But producers are interested in how they can adapt feeding strategies to exploit these mechanisms. One of the first steps is to identify the mechanisms that are driving this compensatory growth response. Defining these mechanisms is useful for determining and identifying biomarkers that can aid in developing strategies for improving growth or identify new criteria on which to base selection for breeding purposes. To address these issues, we had 2 groups of fish that were fed differently. One group was fed continuously, while the other was feed deprived for 1 week and then refed to satiation for an additional 4 weeks. We monitored growth and feed intake during this period. We also harvested liver and muscle samples throughout the study to characterize how physiological mechanisms respond to feed deprivation and short and long-term refeeding. So looking at the graph on the left, you can see that we did not achieve a true compensatory growth response because the body weights of the 2 groups did not converge upon refeeding after feed deprivation. But we can call this recovery growth because when we express these data in terms of specific growth rate, which is the graph on the right, the specific growth rate is the percent of body weight gain per day. You can see that there was a recovery growth in the treatment group that was initially feed deprived. These data indicate faster growth rates in this treatment group, and so we can use these fish to determine what mechanisms might be driving this response. There are 3 main proteolytic mechanisms in muscle that all have very different functions. The ubiquitin proteasome system targets specific proteins for degradation. So it represents a fine-tuning approach to control specific mechanisms, and ubiquitin ligases play a key role in this process. There are hundreds of known ubiquitin ligases, and we have 5 in the multiplex that are known to be specific for muscle and important for muscle growth. 3 of those ubiquitin ligases I'm showing in this slide. 2 of them, FBOX25 and FBOX32, which are on the top 2 graphs, appear to have a role in the different growth trajectories, including feed deprivation and recovery growth. Expression increases after 1 week of feed deprivation, and that's characterized by these red arrows. You can see increases in expression compared to baseline during the 1 week of feed deprivation. But within 24 hours of refeeding, expression very quickly returns to either baseline, in the case of FBOX32, where this green arrow is pointing, or it overshoots the baseline, like what we see with FBOX25. Expression of FBOX25 remains below baseline for several weeks, and baseline is what we're calling the levels of the continually fed fish. FBOX32 also exhibits this overshoot response, but it's not quite as dramatic as for FBOX25, and it's only transient since it occurs only 1 week into recovery growth. The gene on the bottom, MERF3, increases during feed deprivation like the other 2 genes, but it takes much longer to reach baseline upon refeeding and only reaches baseline, remaining unchanged from continually fed fish throughout the period of recovery growth. So collectively, these data suggest that the MERFs are likely not involved in recovery growth because expression returns to baseline and stays there, while FBOX22 and FBOX32 likely play a role in recovery growth. Although the role of FBOX25 in muscle has yet to be fully characterized, studies in other cell types suggest it's a negative regulator of proliferation. So reductions in expression during recovery growth may increase the capacity for muscle cell proliferation and growth. In contrast, much more is known about the role of FBOX32, which is more commonly referred to as atrogen-1, and that's the name of the protein itself. Atrogen-1 is also a negative regulator of muscle growth, and the mechanisms are certainly more defined. Atrogen ubiquitinase integrates proteins critical for muscle proliferation and differentiation, including MyoD and myogenin, proteins involved in anabolic signaling like FOXO1 and elongation factor, as well as sarcomere proteins. So increased expression during feed deprivation is likely promoting reductions in muscle growth capacity, while reduced expression during recovery growth supports an increased capacity for anabolic activities and promotes accretion in muscle mass. A second proteolytic system is the autophagy lysosome system, and this serves a very different role in protein degradation compared to the ubiquitin proteasome system that I just described. The autophagy system serves as more of a non-discriminate proteolytic pathway, contributing to probably about 50% of total protein degradation in muscle cells. Like the previous gene, it seems that this system clearly plays a role in the feed deprivation response, with numerous genes within this pathway upregulated after a week of feed deprivation, which is demonstrated by the red arrows. And this really isn't surprising because during feed deprivation, muscle is broken down to provide amino acids necessary for continued protein synthesis and energy substrates. But what we see during the recovery growth period is that most of these genes are reduced to levels below baseline, and that is indicated by the green arrows. This response seems to occur at a much slower pace, which is greater than 24 hours post-refeeding than what was observed for the ubiquitin ligase genes. So this data suggests that downregulation of these autophagy pathways is important for retaining the proteins in muscle, contributing to improved muscle accretion during recovery growth. Unlike those previous 2 systems, the calpain system appears to be regulated very differently because gene expression profiles largely support reduced expression during feed deprivation. And again, this is indicated by the red arrows. On the top left graph is calpain-1, which is decreased during feed deprivation, but at the bottom 2 graphs, which are the calpastatins and inhibitors of the calpains, are actually increased during feed deprivation. We also see that there's no overshoot response like we saw for the previous two proteolytic systems. Once expression levels are changed during feed deprivation, they return to baseline and are maintained there throughout the period of recovery growth. Now, calpains can degrade myofibrillar proteins, and so reduced expression of this system during feed deprivation may lead to the preservation of the important proteins that are required for muscle structure and function. And it certainly makes sense that maintaining the integrity of those proteins during feed deprivation is important. So once we put together these data, we can start to develop a story about how proteolytic processes contribute to muscle loss or muscle growth during feed deprivation and refeeding periods. Reductions in protein synthesis, coupled with increased capacity for protein degradation via the proteasome and autophagy systems, results in reduced muscle growth. But during refeeding or recovery growth, the proteolytic systems, including the ubiquitin proteasome and autophagy, are likely decreased below levels observed in continually fed fish, suggesting that protein degradation is almost shut down to help the muscle rebuild. This process is likely significant for the faster rates of muscle growth during recovery period. Of the more than 30 growth-related genes that we analyzed in the second multiplex in muscle tissue, there were several that exhibited expression profiles that support a role in recovery growth. So examples of those genes I'm showing in the top 5 graphs. Most notable for this response was IGF-1, IGF-2, and IGF receptors, which are the middle 3 graphs. Although I expected these genes to exhibit downregulation during feed deprivation, which is consistent with reduced anabolic signaling in muscle, they were actually unchanged during this treatment period. And you can see from week 0 to 1 with the red bars, the expression remains consistent with continually fed fish. What we saw was in significant upregulation then in just the 4 hours after refeeding, indicating a rapid response that may help drive recovery growth in muscle, and this is indicated by the green arrows. For IGF-1, levels dropped to baseline within 1 week after refeeding, but IGF-2 took 2 weeks to return to baseline levels. Sensitivity to IGF signaling, which is evidenced by increased IGF receptor expression, which is this 4th graph, also seemed enhanced during early refeeding period. So these 3 genes suggest that anabolic mechanisms regulated by IGF are enhanced during recovery growth. Data also suggests that elevated SMAD expression, specifically SMAD2A, which is shown on the left, and 4A, which is not shown here, are also associated with recovery growth. And in the graph all the way to the right, we have MIF5, which is a transcription factor regulating muscle cell differentiation. And it's also elevated immediately upon refeeding, suggesting an increased recruitment of myosatellite cells into the differentiation program, contributing to improved muscle accretion. While several mechanisms in muscle correlate with recovery growth, mechanisms in liver appear to be less relevant for the long-term recovery response. As expected, numerous growth-related mechanisms responded to feed deprivation, and this is not surprising given the central role that liver has in nutrient metabolism and partitioning. It is well known that plasma IGF concentrations are reduced during feed deprivation, and supporting this response is a downregulation of IGF-1, which you can see in the second graph with the middle bars. As expected, we also observed changes in IGF binding proteins, which are in the third graph down, particularly with IGF binding protein 1b that binds to IGF in plasma, and IGF binding protein 4 and 5, which have more of an autocrine or paracrine effect directly in liver tissue. So reductions in IGF production in the liver lead to reduced IGF-1 in circulation, and this reduces IGF signaling in peripheral tissues like the muscle, documenting a systemic effect of hepatic growth mechanisms. As we observed in muscle, there was extensive regulation of SMAD expression, which is the bottom graph, both up and down, indicating an additional level of regulation beyond just post-translational phosphorylation. But what we saw during the recovery growth response in liver was something different than what was observed in muscle. In the muscle, we saw sustained differences in expression during recovery growth. But what we saw in the liver was that most genes responded specifically to refeeding, like the activin receptor and follistatin, which are in the top right graph, IGF-2 and some IGF binding proteins and SMADs, which are in the second, third, and fourth graphs, generally show a very rapid increase in expression immediately upon refeeding. And each of the green arrows points to that response. I should have mentioned before, the red bars are the feed deprivation response, and the dark green bars are the 4 and 24-hour post-refeeding response. So expression very quickly increases, but also quickly then returns to baseline, usually within 24 hours, but most of the time within 1 week of feed deprivation, indicating that these genes probably are most significant for the liver to recover from the feed deprivation response and begin to regenerate that liver tissue. Those genes with the known systemic effects, like IGF-1 and IGF binding protein 1 and 2, do not exhibit responses that sustain into the recovery growth period. And this is important because it suggests that these mechanisms with endocrine effects are probably not important for recovery growth. The signals that are important for recovery growth appear to occur directly in the muscle itself and could result from other systemic signals like increased nutrient availability, but also from signals produced inside the muscle itself. So with all of these data, we have enough that we can start to produce some heat maps that cluster genes together by common response. And these two heat maps are shown for liver and muscle, and the genes that are related to the growth multiplex, or that were included in the growth multiplex that responded to treatments. The genes on the bottom exhibit largely a feed deprivation response that sustains into the short-term refeeding period. The genes in the middle generally represent a short-term refeeding response, while the ones on the top right are more for long-term recovery growth. And so this can be an efficient way of representing changes in expression, since putting many graphs into a manuscript is often not feasible. So I'd like to switch gears here and talk about a second study that was related to steroid hormone effects. One of the issues with rainbow trout is the onset and progression of sexual maturation, particularly in females, is a very powerful regulator of nutrient partitioning. And during the spawning season, muscle flesh tends to get very, very soft. So because of that, rainbow trout are usually harvested as a food fish prior to the onset of sexual maturation. The two images on the top of this slide represent female rainbow trout that are approximately 2 years old. The fish on the left is a diploid that has been stripped of its fully developed eggs, and we get about 3,500 to 4,000 eggs per female, and the mass of these eggs routinely reaches about 15% of total body weight. The fish on the right is a triploid female, and because of the extra set of chromosomes, this fish is unable to develop eggs and is therefore considered sterile. The viscera, which would be the liver and the gastrointestinal tract, have been removed from each fish, and you can see that kind of sitting below the abdominal cavity. Clearly, the diploid female on the left has almost no viscera fat, whereas the fat has continued to accumulate in the sterile triploid on the right. So the fish on the left has been consuming feed to satiation during the several months up to collection of eggs, indicating that the visceral lipids are mobilized despite this high level of feed intake, because there is such a high energy demand associated with gonad development. Now what's interesting is that this fish will break down muscle proteins, but the muscle will continue to gain weight if feed intake is adequate. It's largely unknown, though, whether nutrient repartitioning is caused by elevated steroid signaling, such as elevated estradiol production that is only elevated during sexual maturation, or it's driven by the negative energy balance that's associated with gonad development. So we performed a study to identify the role that each of these variables plays in the nutrient partitioning response that we see during sexual maturation. We started the study with 2 groups of fish that were approximately 19 months old, so the diploid females were actively developing gonads. The diploids were on a standard controlled diet, as were a group of triploids. And then we had a 3rd group that were triploids consuming estradiol at 30 milligrams per kilogram of diet. These treatment diets were fed for 30 days, and by looking at the response of the estradiol-treated triploids consuming the controlled diet, we can isolate the effects of estradiol. But when we compare the estradiol-fed fish to the diploids controls, which also have high levels of estradiol, but more from endogenous production, we can start to isolate the effects of gonad development. So we were actually quite pleased when we measured the levels of estradiol in the treatment groups and found, this is shown on the bottom graph, on the right side of the graph are estradiol plasma concentrations after 30 days of these treatments. And what we saw was that the maturing diploids in the blue bar and the estradiol-fed triploids on the green bar had levels of estradiol that were physiologically comparable. So that these triploids can serve as an appropriate model for the effects of estradiol. We then harvested liver, muscle, and visceral adipose tissue at the beginning and at the end of the study, and we analyzed total visceral lipid content and fillet yield. So using these samples, we can determine how much each tissue compartment has gained or lost and identify mechanisms that seem to be significant for this response. So this first data set indicates how treatments affected the amount of visceral fat as a percent of body weight on the left and fillet yield on the right. At the beginning of the study, there were no differences in visceral fat content between the diploid and triploids, but after 30 days, the maturing diploids exhibited a lower visceral fat index. But estradiol itself in the triploids did not affect visceral fat content. So these data suggest that visceral lipid loss during sexual maturation is not a result of estradiol signaling, rather is driven by the negative energy balance that's caused by gonad development. Now we saw something different for fillet yield. Whereas the diploids continue to gain fillet weight at an equal rate as the triploids, the estradiol-treated triploids actually exhibited a lower fillet yield. So this tells us that estradiol alone has negative effects on muscle accretion in the rainbow trout. But there's something about the combination of sexual maturation and estradiol that helps maintain muscle growth, and this is obvious with the equal levels of fillet yield between the diploid and triploid controls. So we performed the growth multiplex in liver to identify mechanisms regulated by gonad development and estradiol. Clearly, on the top left graph, estradiols, and the other 2 graphs, which are IGF-related mechanisms, are regulated in sexually maturing diploids and estradiol-treated triploids in a similar direction and magnitude. And we can see this with the blue bars, the comparison between the blue bars and the green bars, supporting that estradiol plays a strong role in regulating these genes during spawning because these data sets respond in a similar direction. More specifically, we see expected upregulation of estradiol receptors on the top left graph, which are regulated primarily by estradiol, and this really is not that surprising. What is slightly interesting is that the estrogen receptor beta-2 expression was reduced with estradiol treatment only. And this is something that we had seen before with estradiol injection, and understanding the genes that are responding to this particular receptor is certainly something that we want to look at. It's also well documented that estradiol downregulates the growth hormone IGF axis, so it was nice to see the expected response for those genes. But the binding protein response, which is on the bottom graph, to estradiol and sexual maturation is certainly novel and supports that binding proteins are regulated during sexual maturation, in that estradiol is the dominant signal that's promoting the changes in expression. Since we saw differences in visceral fat accumulation, it was certainly interesting to see how the lipid metabolism genes responded. The genes that are associated with fatty acid synthesis, these genes are associated with fatty acid synthesis, and there are clear differences between the maturing diploids and the estradiol-treated triploids. Now, the pathway on the bottom of the slide represents de novo synthesis of fatty acids and also the conversion of free fatty acids into triacylglycerols, which is how lipids are stored. The expression of genes related to de novo synthesis of fatty acids like acyl lyase, acetyl-CoA carboxylase, and fatty acid synthase were downregulated in diploids and estradiol-treated triploids. And this provides strong evidence that estradiol downregulates the storage of lipids. And this certainly makes sense because in fish, estradiol levels increase during sexual maturation when there is a high energy requirement to support gonad development. But what we only see in the maturing diploids is reduced expression of genes important for converting saturated fats into the unsaturated forms commonly found in triacylglycerols. Now we know that the visceral lipids are being mobilized due to the high energy demand of gonad development, and that only occurs in the diploids. So these genes may be downregulated to prevent the reincorporation of those lipids back into the storage form. In combination with reducing de novo synthesis of fatty acids, estradiol also seems to increase the capacity for beta-oxidation, and that's the breakdown of fats for energy. In both maturing diploids and estradiol-treated triploids, there is a considerable increase in the expression of carnitine palmitoyltransferase 1A, which is the CPT1A, the first gene on this graph. This is the rate-limiting enzyme that shuttles acyl-CoA into the mitochondrial matrix where it's oxidized for energy. However, there was not such a consistent response in the other genes associated with the beta-oxidation, which are found towards the right on the graph, although the general trend was downregulation. And again, this makes sense because during sexual maturation there is a greater reliance upon lipids for energy, and it appears that estradiol is a major role in regulating this response. So as I indicated before, we have PPAR transcription factors in the lipid multiplex, as well as lipid-binding proteins. The regulation of these genes is largely driven by estradiol signaling. And PPAR gamma is a transcription factor that regulates numerous genes associated with lipogenesis, including fatty acid binding protein 4, lipoprotein lipase, and malic enzyme, all of which were also included on the multiplex and were also reduced, supporting a role for estradiol in regulation of lipid-related activities. Now, the response of fatty acid binding protein 3 and 4 were also interesting because these genes were differentially regulated by estradiol. And we know from mammalian studies that these fatty acid binding proteins have specific affinities for different fatty acids, and the expression levels of these genes in mice are correlated with things like glucose tolerance and insulin sensitivity. So it'd certainly be interesting to further investigate how these binding proteins are regulating nutrient partitioning and growth in rainbow trout. So now that we looked at the muscle, where we applied all 3 multiplex assays to examine the response of nearly 90 genes, we actually only found that 6 of these genes demonstrated an estradiol response. Now, certainly there are many more mechanisms that we did not examine, but the relatively small number of genes here suggests that the liver is the central organ orchestrating the nutrient partitioning response. Regardless, those genes that did respond here include follistatin and myostatin, follistatin and myogenin, both of which were downregulated, supporting reduced myogenesis. Several proteolytic genes were also upregulated, supporting increased rates of protein degradation. And collectively, these findings support estradiol-induced reductions in muscle growth, and we did in fact see reduced fillet yield in these fish. So when we put all of these data together, we can begin to generate a picture of how the signals and mechanisms important for nutrient partitioning during sexual maturation in female rainbow trout, dissecting out the roles of estradiol versus energy deficit due to gonad development. So estradiol appears to specifically regulate genes in the liver, which have systemic effects in muscle, although the effects in muscle could be due to estradiol themselves as well. But the blue arrows represent only what we saw in the maturing diploids, and this response seems to be specific for gonad development. Now, after seeing the powerful catabolic effects of estradiol in rainbow trout, we thought it was important to investigate whether environmental compounds that can mimic estradiol have the ability to regulate these same genes in rainbow trout. One of the major research initiatives in the past 2 decades in the aquaculture industry has been to reduce reliance upon fish meal and fish oil that's obtained from capture fisheries. In an effort to increase aquaculture sustainability, plant proteins are being used in aquafeeds at an increasing rate. But plants contain some anti-nutritional factors that may have negative effects on growth performance. And research has made considerable progress in learning how to process these ingredients to avoid some of these problems. One of the potential anti-nutritional factors are isoflavones, which are found at naturally high concentrations in some legumes, including soy, which is one of the more prevalent plant proteins included in aquafeeds. Some isoflavones, like genistein, which is particularly high in soy, can bind to estrogen receptors, and therefore they are called phytoestrogens. So we investigated the effect of genistein in rainbow trout because dietary genistein consumption is quite high, at least when the fish is consuming a soy-based diet. So studies in mammals have identified effects of genistein on several processes through a number of mechanisms. These mechanisms include binding to the estradiol receptor, inhibition of tyrosine kinase, activation of AMP kinase, and PPAR gamma. This has documented effects in liver, adipocytes, and white muscle tissue that can actually produce beneficial effects in mammals. However, few studies have been completed in rainbow trout to determine potential effects of dietary genistein consumption. And it's particularly concerning because estradiol has such strong catabolic effects in rainbow trout, which is different from what we see in mammals where estradiol is actually anabolic. So it's important to determine if genistein is also having catabolic effects. So to address this issue, we injected juvenile rainbow trout with estradiol or genistein at one of 2 doses. The low dose, which is the 5 microgram per gram treatment group, is about what the fish would consume per day if 50% of its dietary protein came from soy, and the higher dose was injected with the goal of just eliciting a response. So we ran the growth multiplex assay in liver and did observe that the high genistein dose produces an estrogenic-like effect. Now the low dietary dose did not produce an effect after 24 hours of exposure. And this suggests that it may be some long-term, while there may be some long-term repeated effects due to repeated consumption over time, we do have to do a growth study to determine if long-term exposure has an effect. So we saw a downregulation of the growth hormone IGF axis, which extended to the binding proteins, especially the IGF binding protein 2B1, which is in the bottom chart. And this is a protein that has systemic effects binding to IGF in plasma. We also looked at some lipid metabolism genes in liver, including those that responded to estradiol in the previous feeding study. And again, most of these genes were regulated in a similar manner between estradiol and the high genistein dose, suggesting that genistein responses result from activation of the estradiol receptor. So certainly these data suggest that a long-term growth response study is important. Now we looked at responses in muscle and observed elevated expression of proteolytic genes, but only in the estradiol treatment and high genistein dose. But again, these findings support a negative effect on muscle growth. So at the end of this study, we identified several genistein-responsive biomarkers that would be valuable for a long-term feeding study, where diets are supplemented with genistein in a dose-response manner. We can then look at those biomarkers, determine if they respond in a feeding study, and whether they lead to reduced growth or nutrient deposition. So this information can be useful for the producer to make decisions regarding whether soy-based products require additional processing to remove genistein, or whether it's essentially a biologically non-relevant compound because it's so low in the tissue and cannot accumulate in tissues. So after we've completed several of these physiology studies, using the multiplex assays, we were able to identify several genes that positively correlate with catabolic responses, suggesting that these genes are growth inhibitors. These genes might include estrogen 1, which is the SBOX32, or various IGF binding proteins, especially those that bind IGF in plasma. So these genes could be valuable candidates for gene knockout studies that eliminate expression and hopefully produce a response on growth. One of the newer technologies gaining considerable popularity is CRISPR-induced mutagenesis. With this technology, a Cas9 protein, which is the green shape here, must be introduced into the cell along with the guide RNA. The guide RNA is designed to be completely complementary to a 20 base pair region of a gene of interest, usually within the first exon. Once the guide RNA binds the target region, the Cas9 protein cuts at a specific location, and this causes a DNA repair mechanism. The repair itself usually introduces an insertion or deletion of base pairs, and this causes a frame shift mutation or premature stop codon, essentially producing a functional protein knockout. So our long-term goal is to target several physiologically relevant genes, but with any new technology, we first wanted to demonstrate whether we could get this to work in rainbow trout. One of the candidate genes that is useful for this type of proof and concept study is tyrosinase. This is a protein that converts tyrosine into melanin, which is the compound that produces pigmentation. So the value of targeting this protein is that we can see the knockout phenotype immediately because the phenotype is essentially an albino fish. We can see it immediately upon hatch and usually a week prior to hatch once the eyes begin to develop, because an albino fish has essentially white or pink eyes. Rainbow trout are typically 3 millimeters or so in diameter, so injecting the Cas9 and guide RNA into freshly fertilized eggs is fairly crude and can be completed using a handheld microsyringe like you see on the right. So when we tried this, we were pretty excited at our results. We did successfully generate tyrosinase knockouts using CRISPR technology. Although, as expected, we did end up with about 75% mosaics of varying degrees. About 25% of the fish were very albino, and several completely lacked pigmentation, even down to having a pink eye. This phenotype has persisted into adulthood. You can see the pictures on the left, which are the close-up of the eyes, are the sac fry immediately upon hatch. And the fish on the bottom at this point are about 9 months old. So our goal is to make sure that we can achieve germline transition and produce triploids as well. So these fish were produced last year, and our breeding population is starting to produce eggs now, so we are targeting our genes of interest in the next month. Although we are confident from the phenotype data that we have mutant genotypes, it's still important to characterize the mutations in these fish. The GeXP has been a very useful tool for this approach. Because it can do Sanger sequencing, we characterize the various indels, which is an insertion or deletion, just a short phrase to encompass both, to characterize the indels that we found in 2 fish. So fish have 2 tyrosinase genes, tyrosinase 1 and tyrosinase 2, and only the tyrosinase 2 knockout produced an albino phenotype. The CRISPR technology is not always 100% efficient, since numerous fish have the mosaic phenotypes. So with the genotypes here on the top, you can see that we achieved a range of mutations, some of which likely did not affect the functional protein, such as the fish that's shown on top. We have some mutations that are characterized as -6 base pairs, which is a 6 base pair deletion, or a 3 base pair deletion, and this probably does not affect the functional protein. While others, mutations, likely cause a frame shift mutation, that's really any deletion or insertion of base pairs that is not a multiple of 3. Now the problem is that sequencing all of the possible clones from every fish gets expensive and it gets time consuming, especially when hundreds of eggs are injected with the goal of keeping only 50 or so functional knockouts for subsequent phenotype analysis. So we have found a simple and inexpensive way to analyze the extent of mutagenesis which utilizes the fragment capacity of the GeXP. So CRISPR-induced indels occur between the addition of, for example, 5 base pairs to the subtraction of anywhere from 1 base pair to 20 base pairs. So a PCR of the region surrounding that target site will produce amplicons of varying lengths. These amplicons are usually not visualized well using agarose gel electrophoresis, but with capillary electrophoresis, we can very easily separate these amplicons that are very closely similar in size. So a forward primer can be ordered with a fluorescent tag, and that means that we can then detect this amplicon after its separation on the GeXP, much like the technology is applied in the multiplex PCR assay. Using this approach, we can easily screen the fish using DNA that is isolated from a non-lethal fin clip to determine whether that fish is a candidate for functional knockout, given that really any potential phenotype could not be immediately obvious upon hatch. So this chromatogram here represents a wild-type fish. There are two peaks representing two amplicons, one for tyrosinase 1 and one for tyrosinase 2. So we had essentially four primers in this multiplex, a forward and reverse for the tyrosinase 1 and a forward and reverse for the tyrosinase 2. So we get two peaks indicating the, of course, intact protein because this was a wild, or intact gene because this was a wild-type fish. This next chromatogram is from a fish that had the tyrosinase 2 knockout, and the phenotype and genotype are shown at the top of the graph. We can easily identify from the chromatogram that the fish exhibits extensive mutagenesis because each peak represents a particular indel. And we can even characterize the number of base pairs inserted or deleted, which increases confidence that a frame shift mutation did or did not occur. You can see with the red arrow, this would be representative of the intact gene, although we don't know whether that is the fully intact gene or perhaps one gene or one base pair was removed and another inserted in its place. But you can see on the left we have a peak corresponding to a 3 base pair deletion, perhaps a 4 base pair deletion, and clearly an 8 and a 12 base pair deletion. Whereas when we look at the insertions, we have a 5 base pair and a plus 16 base pair insertion. And some of these you can see with the genotype analysis on the top. So for this fish, we clearly have some genotypes that weren't captured in the clones above. So this approach, if anything, provides a more comprehensive overview of mutant combinations. So again, here is an example of the tyrosinase 1 knockout with the intact tyrosinase 2 present in this PCR. Here we appear to have 6 different amplicons, suggesting that this fish has extensive indels, but the fact that we have a -3 and a -6 amplicon suggests that there are probably still appreciable levels of a functional protein being expressed, because that would really only represent a single or double amino acid subtraction from the actual protein. So the next slide, again, reinforces the correlation between the number of mutations observed by the GeXP fragment analysis and the phenotype. These fish we injected for a double knockout, targeting both tyrosinase 1 and tyrosinase 2 by co-injection of the gene-specific guide RNAs. The wild-type fish is all the way on the right, as is the chromatogram for this fish, again indicating the tyrosinase 2, which is the left product, and tyrosinase 1, which is the right product. As we move left with the fish, there is an increasing severity of the phenotype with increasing intensity of albinism. And the height of the peak corresponding to the intact gene, which are on the bottom graph, the asterisks indicate the intact gene. The size of that peak decreases and essentially becomes minor or completely non-detectable in the fry with strong albinism, which is essentially fry A and B. So normally this type of genotype screen is completed using a T7 endonuclease assay followed by polyacrylamide gel electrophoresis. But this approach using the GeXP is much faster, much accurate, and certainly quantitative. So in summary, just to review everything I've talked about today, we've used the GeXP instrument in a number of diverse ways, from gene expression profiling to genotype analysis, and collectively we've made considerable progress in our understanding of how physiological mechanisms regulate growth, nutrient retention, and muscle accretion. We published numerous assays and certainly continue to develop assays that are important for things like egg quality, immune function, and biomarker development. And it's been a real workhorse for us. We've been very pleased with it, and I'm certainly excited to continue using it in the future. And I'd like to acknowledge those collaborators, lab technicians, animal caretaking staff that all have essential roles in completing these studies and taking care of the fish. But also some of the SciEx technical support crew as well, who's been helpful with troubleshooting and just developing and tweaking some of these assays to serve my needs. And if we have any time for questions, I would certainly love to entertain those.
Well, thank you for that informative presentation, Dr. Cleveland. Before we get started on the question and answer session, I would like to remind our audience how to submit your questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Dr. Cleveland, our first question is, which housekeeping genes do you use in the gene expression study, and what selection criteria do you use for those housekeeping genes?
The four housekeeping genes that we use or that we have included in each multiplex is beta-actin, GAPDH, an elongation factor, and also PPIA. Now, we don't always use those genes to normalize. We put the data for all four through Genome, and we find the 2 or 3 most stable and use those as normalizers. Now, one of the advantages of the GeXP is that because you have all the information for 25 to 40 genes, even though there may be a gene that's important for a particular mechanism, it may not change. And so that introduces the concept of using some of the genes of interest as a housekeeper. For example, in our protein degradation plex, we have ubiquitin. And ubiquitin never seems to change. And so we're looking into using that as a housekeeper as well. So we have 4 built in there, but there also are opportunities to go to others that clearly are not regulated by treatment.
Excellent. Thank you for that. Our second question asks, in the analysis of phytoestrogen response in genistein effect on metabolism, how do you measure the response for decreased inflammation and improved insulin resistance?
So the inflammation we could do using our immune-related multiplex, which we have not done yet, but it's certainly gene expression could be used to look at the inflammation response. But we could also look at the abundance of proteins or tissue damage, and that would have to be done in the fish itself. Insulin response or glucose tolerance is something that, while some gene expression, like the expression of GLUT transporters, would be valuable for that, the best way to measure it is changes in plasma glucose over time. So that would probably be something that we would do with fish that have been consuming genistein in a, for example, a long-term feeding study, rather than those that have just received the 24-hour injection treatment that we did here.
Okay, looks like we have time for one last question. That question asks, what delivery mechanism do you use for CRISPR mutagenesis, and at which stage do you induce the mutation?
So because rainbow trout eggs are 3 millimeters or so, we can use a dissecting microscope and a syringe, and if you have a steady hand, which I do not, you can deliver the genetic material straight into the egg. We inject the eggs between 3 and 7 hours post-fertilization, and that's when the egg is still in the single cell stage at about 8 hours. And again, it all depends on the temperature the eggs are held at, but we hold them at 10 degrees, and about 8 hours as well is when we have cell division, so that's 2-cell stage. So as long as we can deliver the Cas9 and the guide RNA before that, we have an increased chance of getting a functional knockout and less combinations of indels.
Thank you. I would like to thank you once again, Dr. Cleveland, for your presentation. Do you have any final comments?
No, well, just to simply reach out to those who are interested in this technology. I'm certainly always available to answer any questions or provide any guidance that any folks will have. If there's interest in any of the publications, I can certainly send you my copies. So please ask any questions you have after the talk via email if you have any.
Thank you. I would also like to thank our sponsor, SciEx, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through June 2017. You will receive an email from Labroots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you, and we will see you next time here at Labroots. Goodbye.